Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 Center for Mesoscale Mapping, housed in the Martinos Center. It is our great pleasure today to have Dr. Ksenia Kastanenka for giving us BrainMap talk. Ksenia Kastanenka is an assistant professor at Mass General Brigham and Harvard Medical School, where she runs a research laboratory with emphasis on sleep and Alzheimer's disease. Ksenia received her PhD in neuroscience from Case Western Reserve University in Cleveland and completed her postdoc training at Mass General Brigham and Harvard. Currently, her laboratory focuses on circuitry disruption underlying sleep during Alzheimer's progression. She has developed a line of work applying optogenetics and multiphoton microscopy to dissect the role of sleep-dependent brain rhythms play in the etiology and progression of Alzheimer's disease. I would like to remind our audience to address your question at the end of the talk by raising your hand or in the Q&A box. Without further ado.
Thank you very much. It's great to be here. My name is Ksenia Kastanenka. That's Ksenia, K is silent for those of you who haven't met me yet. And I'm an assistant professor here at Mass General. And the title of my talk today is Probing Circuit Dynamics by Cell Type in Mouse Models of Alzheimer's Disease. Yeah. Okay, so here at Mass General, we think a lot about the delicate balance between health and disease. What are the elements that tip the scale towards disease, and how can we reverse these? The disease of interest today is Alzheimer's disease. It is a major progressive neurodegenerative disorder that is a major cause of dementia. It is characterized by deposition of amyloid plaques, which are extracellular inclusions composed of amyloid beta, intracellular neurofibrillary tangles that are composed of hyperphosphorylated tau, and neurodegeneration, which is basically cell death that can be appreciated when we compare a brain of an Alzheimer's patient to that of a control individual. In addition to these three major hallmarks of Alzheimer's disease, more subtle changes were recently elucidated, and those include neuroinflammation or activation of microglia and astrocytes, the non-neuronal cells, and disruptions of neuronal calcium homeostasis. For those of you who are not neuroscientists, neurons maintain their calcium levels within a very tight range, about 100 nanomolar. Yet in the case of Alzheimer's disease, we see these significant calcium elevations, what we call calcium overload. So there's this small yet vulnerable neuronal population that we find in the cortex specifically that exhibit these calcium elevations. And we're going to talk about how we use this calcium overload as a functional indicator of treatment efficacy. In addition to memory impairments, Alzheimer's patients exhibit sleep disruptions. They have difficulty falling asleep and staying asleep. And here's an activity pattern of one of those Alzheimer's patients who was particularly active during the hours of the night. So what happens is Alzheimer's patients have difficulty falling asleep. They spend more time awake and less time actually sleeping. And they actually have difficulty maintaining their sleep. So their sleep is highly punctuated by frequent awakenings. And here's a review paper that compares the healthy sleep-wake staging in a healthy individual to that of a control, to that of an Alzheimer's patient. So as you can see in Alzheimer's patients, they spend less time sleeping in general during REM, non-REM stage 1, 2, and non-REM stage 3 slow wave sleep. So I want to draw your particular attention to this deep, slow wave sleep, because that is when memories are consolidated. And since Alzheimer's patients spend less time in this deep non-REM sleep compared to a control individual, Alzheimer's patients have fewer opportunities for memory consolidation that we think leads to their dementia. Slow oscillation rhythm is part of sleep cycle. It was originally coined by Mircea Styriad and was later quite characterized in quite detail by Igor Timofeyev in Canada. Slow oscillation is a brain rhythm that alternates between up and down states at very slow frequencies, about 1 hertz in humans. Slow oscillations can also be monitored in the cat. And what we do in my laboratory is we monitor slow oscillations using voltage-sensitive dyes. Voltage-sensitive dyes, when applied directly to the brain of a mouse, convert a change in voltage into change in fluorescence. So for those of you imaging buffs, we can visualize these waves of activity, slow oscillation, propagating across the cortical mantle of a mouse. So that's what we do in my lab. We load mouse cortices with voltage-sensitive dyes or genetically encoded voltage indicators to monitor this slow brain rhythm in the mouse. And we see that healthy mice have beautiful, robust transitions between up and down states. So now let's talk about the circuit that drives slow oscillations. Slow oscillations are driven by excitatory neurons, shown here in green, inhibitory neurons, shown here in red, and astrocytes also partake in that circuit activity. So what actually happens to the circuit in the course of a single wave of activity, a single slow oscillation? So here is a representation of a network status, excitatory neurons, inhibitory neurons. And their activity is actually very spontaneous. Let's think about layer 5, cortical excitatory neurons would spontaneously fire. And then they would maybe activate their neighbor until the network is able to reach a threshold. And then all of the cells get activated. So at that point, the network threshold is reached, and we get depolarization of a membrane potential of a lot of excitatory neurons. That activates connecting inhibitory neurons, which then brings this episode down. And that sets a period of network depression during which another episode of activity is not elicited. So hence we have these transitions between up and down states. Are you with me? Great. Astrocytes also participate, and we will talk about that in a little bit. So we monitored slow oscillations in our healthy mice or non-transgenic controls and as well as a mouse model of Alzheimer's disease. The mouse model we use is ABPPS1 mouse model. It harbors the Swedish mutation in the amyloid precursor protein gene and the delta 9 mutation in the presenilin gene. So what happens is this mouse model deposits amyloid plaques around 5 to 6 months of age. These mice do not have neurofibrillary tangles, and they have little to no cell death. What's also interesting about these mice is they actually start producing oligomeric A beta that we think are the toxic species very early, about 3 months of age. So that's something to keep in mind. So we monitored slow oscillations in this mouse model, and we see impairments of slow oscillation rhythm. So then we wanted to see what is it about the slow oscillation that is impaired. We performed Fourier transform analyses that told us the power frequency relationships, and we saw that the power, which is directly proportional to the amplitude of the wave, was significantly lower in our AD mice. And that happened very early, starting at 3 months of age. So starting at 3 months, when there's a oligomeric A beta that is impairing circuit activity, we see decreases in the power of slow oscillations in our AD mice, and that continues into plaque-bearing years. The frequency of slow oscillations is maintained, however, at around 0.6 hertz. So that told us that APP mice basically exhibit slow oscillations, albeit at lower power or at lower amplitude. So then we wanted to figure out whether it is oligomeric A beta that is impairing the slow oscillation power or it is an artifact of the overexpression transgenic model of Alzheimer's disease that we're using. So we would take a non-transgenic wild-type mouse with robust slow oscillation power, and then apply a transgenic condition media preparation. So this preparation is enriched in A-beta oligomers, and we see a drop in slow oscillation power, very similar to what we saw in our transgenics. So the power went down while the frequency was maintained. So that told us that it is the A-beta, and specifically oligomeric soluble A-beta, that is decreasing the slow wave power and disrupting slow oscillations. So then we wanted to take the circuit mechanism exploration a little bit further, a step further, and wanted to see what is happening to the circuit at these early stages of Alzheimer's disease. We would take an APP mouse with low power of slow oscillations, and then we applied GABA or inhibitory neurotransmitter directly onto the brains of these animals, and we saw rescue of slow oscillation power. So that told us that the circuit is hyperactive at these early stages of Alzheimer's disease because GABA was sufficient to rescue. Okay, so now that we saw that slow oscillations are impaired very early in Alzheimer's disease in this mouse model, the next question was, can slow oscillations be restored by optogenetic targeting of different cell types? The cell types we used were excitatory cortical neurons, inhibitory cortical neurons, or cortical astrocytes. The reason we wanted to do that was to try to get at the trying to dissect the circuitry underlying the impairments of these slow oscillations. Maybe there is a specific cell type whose activity is impaired, and that leads to the impairment of these slow oscillations. And then if so, if we can rescue slow waves by targeting these different neurons, can optogenetic restoration of slow waves actually slow AD progression in our mice? And that was another really critical question to answer, because we wanted to see if perhaps the impairment within the circuit was causal for Alzheimer's progression and not simply symptomatic of disease or an epiphenomenon of the disease. Because if slow wave impairments actively contribute to Alzheimer's progression, then we can target those therapeutically in patients. Okay, so we carried out a couple of studies with two aims. Aim one was to determine if optogenetic treatment restores slow oscillations in APP mice, and we used voltage sensors to monitor slow oscillations in our animals after targeting of these different neuronal types. And aim two was to determine if optogenetic rescue slows AD progression in our animals. And here we would monitor amyloid plaques through cranial windows using high-resolution multiphoton microscopy. And we would monitor neuronal calcium levels with our genetically encoded calcium indicator called yellow chameleon, and I'll talk about a little bit more. So let's talk about the first study. As I mentioned, we use optogenetics. Specifically, we targeted channel rhodopsin, which is an excitatory opsin that looks something like this, into either excitatory neurons, inhibitory neurons, or astrocytes within anterior left cortex. So a single injection. And we would shine brief flashes of blue light at the normal frequency of slow oscillations, about 0.6 hertz. And then with brief flashes of blue light, we would see membrane potential depolarization or firing of the neuron, or astrocytes don't fire, but it would depolarize their membrane. So with these brief flashes of blue light, we were hoping to drive the cell activity as well as elicit slow oscillations. So let's talk about optogenetic targeting of excitatory neurons that was injected here. We saw beautiful injection of cherry.
Yes, may I ask a question?
Sure.
Could you talk about just very briefly, the different ways to, alter activation of neurons? Maybe to a genetic version. Other people use the chemicals, the drugs. Could you just make a sentence or two, like how you decided on which strategy to use?
Yeah, so that's a fantastic question. So basically the question is, there are many different types to activate neurons, for instance, now. So the difference between optogenetic and chemogenetic activation, for instance. So I think it depends on the application. It depends on what you're trying to achieve. So optogenetic allows millisecond precision, so very fast activation of neurons. So for instance, here, we are targeting inhibitory interneurons like parvalbumin, for instance. Those fire very fast. So if we wanted to drive their activity at, let's say, 40 hertz, we could do those with some of the opsins, right? So we could activate these cells at very fast frequencies. So very precise temporal resolution. Chemogenetic, for instance, doesn't do that, right? You apply a drug and then the neuron is activated. So using chemogenetics in this case probably wouldn't be as effective, although there was another laboratory that tried to activate thalamus with chemogenetics, and they could rescue slow oscillations as well. So the another advantage of targeting with chemogenetics, it's a more translational technology, right? Applying a drug versus, installing a fiber optic fiber, right over the cortex with optostim. So optogenetics is not as translational, chemogenetics is, but there's this, drawback of temporal resolution. So it really depends on the application.
Thank you.
You're welcome. Okay, so what we figured out with excitatory neurons, that with these brief flashes of blue light, we were able to elicit slow oscillations, which was really cool. So we would excite the neurons here, and we would actually monitor slow oscillations in the contralateral hemisphere. So the only way that power would go up and transfer all the way to the contralateral hemisphere if the wave was generated and then would propagate all the way to the contralateral hemisphere, which is what these waves do, which was amazing. And then as soon as we turned the light off, the slow wave resumed its disrupted state. So we were able to increase the power of slow oscillation with light activation while maintaining the frequency. Can we do the same thing with inhibitory interneurons? So here's a picture of Jack Zhao. So the excitatory stimulation was actually something I did when I was a postdoc here at MGH, and a lot of you know I started here. And then when I started my own lab, Jack was one of the first postdocs in my lab, and he said, I'm just going to try exciting inhibitory interneurons. So he targeted an excitatory opsin into an inhibitory interneurons by breeding GAD-CRE mice with our transgenic mouse model of Alzheimer's disease, ABPPS1 mice. And his channel rhodopsin here was CRE dependent. So he was able to target inhibitory neurons in the presence or absence of amyloidosis. Okay. And then again, with brief flashes of blue light, Jack was able to power. We target inhibitory neurons, we get increase in slow oscillation power. Whoa, how about astrocytes? We were on a roll. Let's just test it, right? So why were we actually interested in astrocytes? So astrocytes have been traditionally known as support cells for neurons to provide nutrients and take out waste as a result of neuronal activity. However, recently, the work of Alfonso Araki and colleagues showed that astrocytes are actually active participants in circuit function. There is a communication between neurons and astrocytes. Astrocytes respond to neurotransmitters released by neurons, and astrocytes release gliotransmitters. So they're actually participating in circuits, as shown here. So here's an astrocytic process that is working together with the presynaptic and postsynaptic neuron, and that results in oscillatory activity. And there was a paper at the time, by the time Evelyn Lee joined my laboratory, graduate student, and there was a very clear data showing that optogenetic activation of astrocytes actually can elicit slow oscillations. And that the calcium transients within astrocytes and neurons are actually coincident, right? There were a couple of GCamp and RCamp, and they saw that the transients of these slow oscillation transients are very in sync between astrocytes and neighboring neurons, which was really cool. So Evelyn decided to target astrocytes with optogenetics as well. Now, remember, astrocytes don't fire action potentials, but we believe that by flashing blue light at the frequency of slow oscillations, we could actually depolarize the membrane of astrocytes, and somehow either by targeting the circuit neurons or through astrocytic gamma junction, we get propagation of slow oscillations. Because Evelyn saw an increase in the power of slow waves when she optogenetically targeted astrocytes at the frequency of slow waves. Okay, so conclusions from AIM-1, slow oscillation rhythm is impaired in APP mice. A-beta disrupts it while GABA restores it because circuit is hyperactive. Optogenetics can be used to manipulate the power of slow waves. Optogenetic targeting of excitatory neurons at the frequency of slow waves restores slow wave power. So does optogenetic target of inhibitory and optogenetic target of excitatory of astrocytes as well. So that told us that it doesn't matter really whether we're optogenetically activating excitatory, inhibitory neurons, or astrocytes. As long as we rescue the circuit, the slow oscillation gets rescued and propagates. Or does it make a difference on disease progression? So that's the next question, right? So in this case, we would optogenetically target, again, excitatory inhibitories or astrocytes and see, does it make a difference on AD progression? The way we monitored AD progression in our animals is by looking at amyloid plaques through cranial windows and monitoring calcium levels within neurons, as well as several other readouts that I will describe. So here is this long chronic experiment that we would carry out. We would use young APP mice, target channel rhodopsin to excitatory inhibitories or astrocytes. We would also target a virus with the yellow chameleon 3.6, which is our ratiometric calcium sensor that allows us to determine the basal levels within neurons. We would perform craniotomies, perform voltage-sensitive dye imaging to make sure that slow waves are impaired, install a fiber optic cannula, and then perform a chronic light treatment. The chronic light treatment would be at 0.6 hertz for 24 hours a day for 2 to 4 weeks. So continuous stimulation. And I will explain why it was continuous in a little bit. And then we would inject methoxy, which would cross the blood-brain barrier and bind to amyloid plaques in the brain. And we would image those plaques with multiphoton microscopy during imaging session 1, and then some of the animals would be re-imaged. We would image amyloid plaques and the calcium within the neurons. And the working hypothesis was that chronic treatment with light activation of channel rhodopsin would restore slow oscillation in APP mice and lead to slower plaque deposition, as well as normalization of calcium. Okay, so just to orient you a little bit again, this is where channel rhodopsin was injected and the fiber optic cannula. So that's where light was delivered. This is where yellow chameleon was injected, our calcium sensor for neurons. And also this is where slow waves were monitored with voltage sensors. And this is where we monitored amyloid plaques. So by activating these neurons or astrocytes, we would expect a wave to propagate into the contralateral hemisphere and rescue the circuit that would have beneficial effects in our AD mice. Does that make sense? Great. Okay, so let's see what happened to plaques. So we would monitor amyloid plaques in 6-month-old animals that were not treated with light activation of channel rhodopsin of excitatory. So these are excitatory neurons, or treated. And you can see in our treated animals that we saw fewer amyloid plaques compared with untreated animals. So number of plaques formed was significantly higher in APP mice compared to the treated animals, we actually saw a little bit of clearance. And at this point, we were not sure whether it was clearance, and I will tell you a little bit later that this probably was. And amyloid plaque burden change was also significantly higher in the untreated animals compared to those that were treated. And then this is the data from inhibitory neurons that we published with Jack. He saw that optogenetic activation of inhibitory neurons led to fewer amyloid plaques compared to untreated animals. The plaque burden was significantly lower. So what happened to plaques? Are we getting fewer new plaques being deposited? The answer is probably yes. Are we getting clearance of amyloid plaques? And the answer is definite yes. Because Maggie Macy, who was working with Jack at the time, went and counted. She first stained microglia with IBA1 marker, and then she went and she counted microglia around amyloid plaques and away from amyloid plaques. And they found that optogenetic treatment resulted in an increased number of microglia. Microglia cell body volume went up and process length went down. So there was a morphologic change in microglia, suggesting that microglia might be becoming more phagocytic. When they're phagocytic, their cell body swells and their process length shortens. And plaque-associated microglia number also went up. So there were more microglia or reactive microglia, I should say, around plaques. Jack also did a flow cytometry experiment, and he saw that, yes, indeed, microglia plaque number did go up, and also their microglia had methoxy inside of them. So there were more microglia with methoxy-positive amyloid inside of it. So that told us that optogenetic targeting of inhibitory neurons, in this case, leads to plaque clearance because we get more microglia that is phagocytosing amyloid. So hence increased clearance. Okay, so now let's talk about yellow chameleon that, again, was injected over here. Okay, so how does this work? So for those of you who are aware or familiar with GCAM, that has a single fluorophore and allows us to determine the relative changes in calcium, yellow chameleon works a little bit different because it has two fluorophores. It is a Fred-based probe, and when calcium binds to it, it brings the two fluorescent proteins together, and that results in Fred. So we get activation of CFP, which then activates YFP, and hence the ratio of YFP to CFP is significantly higher with high levels of calcium. So if calcium is low, these guys are far apart, there's less Fred, and the ratio of YFP to CFP is lower. So high YFP to CFP numbers are directly proportional to the basal calcium concentration. High levels of calcium and then low ratio levels correspond to lower calcium levels. And we can actually color code those high calcium levels and low calcium level expressing neurons here in this picture, for instance. So neurons with high levels of calcium are shown in red. So these are the sick neurons, and the blue neurons are healthy. And then previous work from a PhD student and then postdoctoral fellows in the laboratory of Brian Baskey, where I did my postdoctoral training here at Mass General, determined that it is amyloid beta that elevates intracellular calcium, right? So APPPS1 mice have this calcium overload or a few neuronal processes that are red or yellow, okay? And then Michal and Eloise showed that if they were to take a healthy mouse and apply transgenic conditioned media, this preparation, which is enriched with A beta oligomers, we get calcium elevations as well. So we know it's oligomeric A beta that results in these calcium elevations. So can we fix it? Can we fix it with light activation of channel rhodopsin targeted to either excitatory neurons, inhibitory neurons, or astrocytes? The answer is yes, we can. So here's some brains or some images, multiphoton microscopy images you're looking at. So if we were to rescue slow oscillations by targeting, this is inhibitory neurons, we don't see that many neuronal processes with calcium overload. So you don't see that much red. However, neurons that lacked, or mice that lacked treatment, had these neuronal processes with calcium overload. So calcium overload was significantly lower in treated mice compared to those that were not treated. And then we wanted to look at sleep. So slow oscillation is a sleep brain rhythm, right? So what happens to sleep? And it took us a while to get it done, but we got it done. So Jack used a fully implantable EEG, EMG sleep telemetry system to monitor sleep-wake cycle in his animals. He was able to distinguish between non-REM, REM, and wake. For those of you who are not sleep experts, I'll explain. So non-REM sleep is characterized by high amplitude, low frequency oscillations in the EEG, little to no EMG. REM is characterized by low amplitude, high frequency EEG and little to no EMG. And wake is characterized by low amplitude, high frequency EEG and a lot of EMG, suggesting that the animal is moving. So remember, non-REM sleep, that deep, slow wave sleep, that's when memories are getting consolidated. So that is the sleep stage that we were particularly interested in. So normally, healthy non-transgenic mice, they spend time sleeping and awake during light and dark phases, which was the reason why we decided to drive light activation of channel rhodopsin 24 hours a day, because we were not sure when they would be sleeping and we wanted to potentiate that sleep. We were not sure when they would be awake and we wanted to make sure they're sleeping during that time. So we decided to drive the slow oscillation rhythm for 24 hours. So as you can see here, healthy animals spend more time in slow wave sleep. So there's more purple during the light compared to the dark. Same in APPs, animals spend more time sleeping during the light compared during the dark. However, the amount of sleep, the amount of time APP mice spent sleeping was significantly less, and they spend more time awake. So our APP mice spend less time sleeping and more time awake. What does that remind you of? Alzheimer's patients. That's exactly what happens in Alzheimer's patients. So we were very excited that we modeled sleep impairments, if you will, with our mouse model. So now can we target inhibitory neurons with light activation of channel rhodopsin, rescue slow waves, and see if it rescues sleep? That was the question here. And interestingly, optogenetic stimulation increased the amount of purple. So there's way more purple here than here. Actually, more purple everywhere during the light and dark phase, as it turns out. So optogenetic targeting of inhibitory neurons rescues slow waves and increases non-REM sleep, as well as decreases wake in our mice. So that was really amazing that we could restore sleep in our animals. Was that what happened with optogenetic targeting of astrocytes? And the answer is yes, surprisingly yes. So again, we saw more purple during the light and dark phases in our treated animals compared to those that were not treated. So the amount of time animals spend in non-REM went up, and the amount of time they spent awake went down, and that was significant during the light and the dark phase as well. Okay, so we can rescue slow waves, we can rescue sleep, we can decrease amyloid burden, we can rescue calcium homeostasis within neurons. How about memory? How about dementia, right? Alzheimer's is a dementia. How about memory impairments? So mice don't have memory impairments, right? Like healthy mice don't. And if we were to put them into a fear conditioning paradigm, you know, the non-transgenic animals, they do pretty well on this test. So let me tell you what the test is. So we would inject chanorodopsin and then allowed it to express. And then in this case, Evelyn put the mouse into, on day 1, into a fear conditioning chamber where animals would be exposed to a foot shock. And then she would put the animals back to its home cage where animals would experience light activation of chanorodopsin targeted to astrocytes. So their slow waves would be rescued while animals are sleeping and consolidating that fearful shock memory. The next day, Evelyn would put her mice into the same chamber, yet in absence of foot shock. As it turns out, healthy wild-type mice, they remember where they received the foot shock, so they freeze, right? They don't like being there, they freeze. And as it turns out, APP mice, they don't remember the chamber very well, so they don't freeze as much. So their freezing is significantly lower. However, if we rescue slow waves by targeting astrocytes in this case, we can rescue that freezing behavior. So that suggests to us that optogenetic rescue of the circuit rescues the memory consolidation, sleep-dependent memory consolidation as well. Does restoring slow oscillation rhythm ameliorates AD progression in APP mice, which is really exciting to, to report, and would be great to hear for those of you who are working with humans. How can we translate this to humans? How can we translate this to patients? That's what I would like to, open for discussion. Just so really quickly, conclusions: restoring slow wave power halts amyloid deposition, restores calcium homeostasis within neurons, rescues sleep, as well as memory. Thus, targeting slow wave activity in AD patients might prevent neurodegenerative phenotypes and slow disease progression. However, maintaining the right frequency is really important. And I told you that we're activating all of these different cell types at 0.6 hertz. If we were to drive slow wave frequency at twice the rate, 1.2 hertz, things actually get worse. So the frequency is really important, not the type of cell that we're targeting, as long as it's within the circuit. So here is a schematic from a review paper we published with Evelyn now in 2020. We're working in an updated version with 3. So slow wave sleep, specifically deep non-REM slow wave sleep, is impaired in AD patients as well as mouse models. Slow wave activity, that impairs slow wave activity, lymphatic clearance, which I didn't talk about today, as well as memory consolidation, that increases A beta and tau as well. And that in turn further impairs slow wave sleep, clearance, slow wave activity, and increases A beta. So there's this vicious cycle. I mentioned that we can use optogenetics to try to break that cycle by rescuing neuronal activity. Christiane suggested using chemogenetics, which is another great technique. Patients with AD, perhaps we could use some of the non-invasive stimulation technology, or some are a little bit more invasive to, to break that cycle. And I'm open to discussing that. So restoring normal circuit activity patterns is necessary for healthy neuronal function, and when disrupted, leads to disease. And here's my lovely lab. I'm so proud of them because they did all the work and doing a lot of the work that actually the talk I gave today provided the foundation for their work. So I'm really proud of each and every one of them. And thanks for funding, and thank all of you for your attention. We'll take any questions you may have.